So by running those molecular markers, I can tell you that this is gelato 41 and the other one is not gelato 41. Okay, so that is extremely important, again, for not only for consumers who are probably they have the right to buy and to consume what they are told, right? But also for growers and for breeders. For breeders, it's extremely important because this is the way to protect their invention. If you are working many years in some particular plant, you want to get some particular phenotypes, you have the right to protect your invention. And with this panel or with these cheap molecular markers, you can do it.
Detect and protect. We can't deny that from an agricultural perspective, cannabis is a plant like any other. That means it's subject to threats from virus, fungi, and other potential crop contaminants. Early intervention is critical. We speak to Fernandez and Ajith Anand from MyFloraDNA to better understand how to mitigate these and other risks. Hello and welcome to the Lobsterpot podcast. I'm Dave Barton, and what's your name again? Jamie B, Jamie Bonthron. How you doing? You all right, sir? How's the today shaking out for you? You a happy person? You certainly look it.
I am. I'm dead. I'm dead chuffed. I need to get this a bit sorted. We get some greenery, which is a good start, but we need to figure out the rest of it because I'm missing the art from before. You know, it's
you need greenery like this.
Yeah, proper greenery.
Well, within reason, because I guess we could, we figure it out. Well, maybe our friends today can help us figure something like that out. That's my awkward segue into introducing our special guests today. We have Fernandez and Ajith Anand from MyFloraDNA based in California. How you doing, guys? Good to see you.
Good morning. Nice to see you. Thank you very much for having us today here.
You're most welcome.
Good afternoon, and a pleasure to be connected with you folks live today, and looking forward to wonderful discussion.
Excellent. So tell me a little bit about what you guys are doing, because again, we're talking around, there's always lots of discussion around, sort of provenance, around the kind of accuracy, around, GMP standards and things like that. But I guess when it comes to kind of DNA and cannabis, how does that come together? And just tell us a little bit about what you're doing, because it's quite interesting. So tell us more.
So let me start. We are a scientific company. As you say, we are located in California, in Sacramento, very close to Davis, UC Davis. This is where most of our scientific team did either the master or PhDs or postdoc. Okay, so we are molecular biologists and geneticists, and our main strength is biotechnology and all the DNA science right behind that. We have been working with many crops as a team, and since 2022 we are focused on cannabis. Okay, all the science, all the protocols, technology that we have learned in academia and other crops, we want to bring that to the cannabis industry. So that is the main goal, the main mission at MyFloraDNA. Bring genomics and DNA technologies for the growers, breeders, tissue culture, and eventually consumers.
Oh, very cool. So again, there's a big focus on, sort of detecting sort of viruses and fungi and things like that. And that's something, and sort of crop contaminants, that's something that your work is, critical to in helping prevent that as well. Can you tell us a little bit more about that? Because again, when we've spoken before, it seems to be a very interesting, and also, we're talking about prevention. We talk about, cannabis itself is a plant, and I think it's almost easy to sort of forget that unless you're actually in the sort of growing space itself. And plants are subject to so many different kind of external factors. But how do you kind of reduce the kind of potential contaminants, and how does that kind of dovetail with what you guys are doing, as well as the kind of genetic stuff as well?
So let me take on that. So plants, like any other organisms, have infestations, right? There are infections that are on any living organism. So like all living organisms, plants also are prone to diseases and pests. So pests and diseases are the biggest two concerns for any producer, whether it is cannabis or whether it's any other row crops or any other vegetables. Now, coming to the pests, there are common pests that we often see, and most of those are insect pests and herbivory, right? Insect pests includes your mites, aphids, spider mites, for example, and then you have aphids, thrips, fungus gnats, and many other pests that are pretty common in plants. Now, when it comes to diseases, most of the diseases that we see in cannabis are caused by viruses, viroid, and fungi. As we look at the market today, and with cannabis being one of the largest crop, sixth largest crop in terms of the value creation it creates, it is economically important for us not only to look at cannabis as a plant which is producing its compounds, which is mostly the secondary metabolites, the phytochemistry. This is like a bioengine. It produces a biofactory, I call it. A lot of phytochemical things that are, that's so much beneficial to both the humans and as well as, it has its recreational value too. So both on the medicinal and recreational value. One of the biggest concern for any cannabis grower is the presence of HLVD, which is the hop Latin viroid. It's a viroid. It's a very small RNA molecule. Like as we were talking about, as a company which is involved in genomics and nucleic acid, every living organism is made up either a DNA or an RNA. In this case, this is an RNA, which is very small, just 256 base pairs. And so it's got a very looped structure, like it has rod-like structure with little bumps and troughs, which I call that. And it is such a small molecule which cannot by itself differentiate or grow in a plant. It cannot replicate. Beauty is that this viroid is able to piggyback on the host machinery to replicate. And that's how this viroid and replicates in a plant. So it infects a plant most often through mechanical, cuttings and not having a great phytosanitary practices. Now the viroid, once it is in the plant, the beauty again is that this can be latent for a long period. It can be dormant and nobody notices it. And when it really expresses the phenotype, then you see a dramatic impact on the plant in terms of vigor, brittleness of the plant, stunting. The flower quality decreases, the phytochemical compounds decrease significantly from 50 to 70%. This includes THC and other cannabinoids. And that leads to a huge economic loss. And from one estimates, it is around $4 billion in the US. And in another estimate that came out in the 2021 was that almost 90% of the cannabis that is grown in California is infected with HIV.
An enormous amount. I think a lot of the time, Dave made a really good point, which is people tend to forget that cannabis is a plant, because as an end consumer, you end up with the final product in whatever form it is, drink, edible, you just buy flower, it doesn't even really resemble what it started out life as. So I imagine a lot of people who might listen to this might not even understand how you'd go about finding out whether or not something was infected. Obviously, that's something you guys take care of. What does that process look like from a high level? So a business presumably sends you a cutting, and then what happens?
Basically, what we recommend to our clients, so we have done a lot of research. Actually, last year we published the largest essay in the field with around 5,000 samples, in order to find out more about this viroid and what is the best way to collect the samples, which plant tissue, and so on. So we recommend to our clients to send us. So basically, when they make a purchase, we send them a box with small tubes, like vials, and the clients, they cut a little bit of root and a few leaf tissues, So, and then they send back to our lab, and with molecular biology, we do the testing, and within 2 days, they get the results in our platform. which is MyFlora Cloud. So it's a very straightforward step and very easy actually. And the beauty of our technology, the one that we developed last year, is that we can analyze several thousand of samples in a daily basis, having the highest accuracy. So that allows the growers to make this first screening of the plants that they are infected or the plants that they are not infected.
And to add to Angel's point, last year we did one of the largest study, and we came up with almost 5,000 plants from one of the largest growers in California. And there has always been a confusion around, what is the right material to test. And we wanted to get into that. We wanted to make sure that we provide our customers, clients, the best services in terms of being able to know, keeping in mind this particular viroid is sometimes dormant, and it can just be spatially distributed in such a way that we don't know which tissue to take, right? So we did a very systematic study. We took parts from the aerial part of the plant, leaves, and we took the petioles, we took the roots, we did a screening of those. And long story short, we say that it is combination of, you want to, touch every point, right?
Yeah.
You just don't want to take some part of the plant where there is no viroid. So our recommendation today, based on a huge study, which, and as Angel mentioned, we published it, and we wanted to make sure that the knowledge we have is shared with the public, is that sample both leaf and root. And right now, all our clients, we recommend them to get material from different parts of the aerial part of the plant, as well as have a small component of the root. And that way, we are able to cover the whole length and breadth of the plant and be able to give you more precise. Our accuracy is around 98, 99%. And keep in mind that COVID virus or any other viruses we have worked with, this is not a virus, but it's a viroid, is that all the detection depends on the titration levels. So what levels of the viroid or the viruses is in the plant, right? And sometimes you do have those, numbers which are not in the very sweet spot.
I was going to say, in terms of, obviously you talked about the sort of business impact and the potential economic impact, or not potential, but actual as well. And talking about some of those, figures of lost kind of, crops and things like that, and obviously lost revenue. To what extent do you feel like a lot of growers kind of appreciate the need for something like this? Do they see it? Do they understand it? If we're talking to a lot of legacy growers potentially, I guess you're pretty close to I don't know how close, but to the Emerald Triangle, where arguably some of the best cannabis in the world is grown, so I'm told. But at the same time, it's are they really understanding this sort of granular approach to understanding not just what's wrong with my crop, but how to prevent it? Is that kind of, do they see the value in that? Is it a difficult sell, or is it really more and more people are understanding that there's a need to kind of analyze plants at this level in order to kind of maximize the yield itself?
So two things. The satisfaction of our client is there because most of them, I would say 95%, they are recurrent clients. So that means that they test either every week or at least minimum every month. So that means that they see a value. And also talking to VIP clients that are probably largest cultivators in the state of California for sure, and even in other states, they come to us and say, Angel, MyFloraDNA team, you save our production. You save our production. So that means that they see a real value. And thanks to those investments in testing, they can screen for the plants that they are infected and get rid of them. So for us, as a scientific company, again, this is the most satisfactory reward, is when a client comes to you and say, yeah, you save our production. Please keep working and keep improving. So anything else to add, Ajith?
Yeah, I think always we say is prevention is better than cure, right? There is no way we can actually eradicate this thyroid. And so always we suggest is mitigation. Mitigation, well, we have seen big cultivators are coming and saying that they had, I mean, we had done the diagnostic, but we have seen like 50% and bringing down them to single digits. And we have recently had another customer which we had 35%, and we have brought them down to again single digits. So a constant, a survey of your plant that is in your cultivation is the only way to, and using this molecular diagnostic. So just imagine of a time when people are changing their mother plants, that they wanted to bring in new mother plants, and the growers are steadily doing that in this business. The best thing is get them certified. And the easiest way to certify is collect the sample and send it to a lab like us, where we can actually give the results within 48 hours, and the plant is quarantined only for 48 hours. And the decision then is up to the grower and the cultivator, whether they want to keep something which is infected and take the risk, or the results is black and white. So you just make a decision. I think what Angel is saying is we are getting so much of valuable input from our clients and customers in terms of appreciation, and see they are seeing the value. So those who have, those who have gone into this process, they have a very standard SOP, and they do meticulously the job that they should be doing, and they're seeing the benefits. They're definitely seeing the benefits of having a diagnostic protocol in the in the group facility. And I think it's word of mouth. We're getting more and more people who are interested. It's about an education, like what you're doing here is exactly what we want. It's an educational effort to also tell people that everybody in this community, cannabis, who are growing cannabis and who are part of a big community today, that it is a two-way traffic. It's a learning for us by studying what you have in your grow facility. It's also providing back the information that we can figure out, we diagnose from your studies. And so that way, there is more awareness and appreciation. This is one of the biggest disease, but there are much, much more to, again, as a plant, there are going to be more diseases, and they need to keep an eye on that.
Yeah. It's not a one and done, it's an ongoing process. Now, presumably you guys being heavily involved in genomics, it's not just the kind of identification of viroids or fungi or anything like that. It's also to do with provenance and, a plant being what someone says it is, which in the world of cannabis is very important. People buy particular strains, whether it's recreational or medical, for particular reasons. I don't know if you can speak a little bit to that side of what you do.
So we developed last year actually also a technology by using DNA molecular markers that they are specific just for cannabis, and allow us to differentiate within cultivar strains. So by running those molecular markers, I can tell you that this is Gelato 41 and the other one is not Gelato 41. Okay, so that is extremely important, again, for not only for consumers who are probably they have the right to buy and to consume what they are told, right? But also for growers and for breeders. For breeders, it's extremely important because this is the way to protect their invention. If you are working many years in some particular plant, you want to get some particular phenotypes, you have the right to protect your invention. And with this panel or with this chip of molecular markers, you can do it. Okay, so it has many winners. So breeders, also the growers, and finally the consumers. So it's like we generate like the passport number or social security number for each single plant. And let me see if I can share with you my screen. Let me see. So we call it certificate of cultivar identification. So this is like the license plate for every single plant. So this is an example, Donny Burger, but you see all those squares. So every single square corresponds to a DNA molecular marker. So this particular cultivar or strain, Donny Burger, the profile is going to be different as other to another cultivar, Gelato 41 or whatever, right? So we are now trying to standardize this technology in all the cannabis goods and also in the cannabis process. So like that, everyone can be sure that what they are consuming is what they are paying for.
Yeah, I mean, it's really interesting, and I think again, we hear a lot of different types of things, different solutions that people are doing this, and I guess it's kind of like once you've kind of got a lot of this data, what other opportunities are there to leverage it? Do you feel like that's something, like additional services that could be, something you can sort of build on top of, or is it something potentially that could be useful to customers long term? I think it's just interesting where you've got this, more and more this kind of collating of data and collection and thinking, okay, it's great, we see the purpose right now, but there's always going to be a future purpose for collecting information of this. How do you see that moving forward?
I mean, Dave, let me be clear here. So are we talking about collecting the data based on DNA fingerprinting? Are we?
Yeah, I think where you've just got lots of information like this, where you're doing all these different tests. I mean, how else could that sort of be leveraged, I guess, in a sort of commercial, academic sense?
For this particular technology, DNA fingerprinting, I just want to make clear that those are agnostics DNA markers.
Okay.
So the cannabis genome of plants in general, they have around 30,000 genes. So that means that there are many, many millions of molecular markers. And we selected around 3,000 markers that they are not associated with any particular trait of interest. Those molecular markers, they are just specific for saying, okay, you are Dave. And
purely to identification purposes.
It's just to differentiate, to separate, right? But they cannot to tell you, oh, this variety is resistant to powdery mildew or is not resistant to powdery mildew. Those 3,000 molecular markers, they are agnostic, not related to any agronomical trait. So, and we did that express. We did that express because in the past there were some conflicts and polemics with other genomic companies. So we wanted to avoid that. We wanted to be completely transparent and make sure that all the clients, they understand that the only information from those molecular markers are just enough to differentiate among cultivars.
But I guess, do you think long term it could help, growers create more robust strains or more, more varied or kind of more resistant to, viroid strains?
That's needed.
So biotechnology has the power to do that, right? And it's not that we are only focused on cannabis. Just look at the whole seed industry that Angel talked about. And I have been in the seed industry 15 years before I came to this, to the world of cannabis. And I'm a plant enthusiast. I always see an opportunity to apply some of this learning and knowledge to a crop that needs more data and science-based solutions, which is the only way to improve the crop productivity. So if you take examples of papaya, banana, and all those, there have been a lot of work that has been done by some of the nonprofit organizations and academic institutes that have really dealt with producing varieties which are resistant to viruses, right? And the same is applicable. So it's an integrated approach of understanding the genetics, molecular breeding, and then introducing biotechnological tools. And that is the only way to get to the solution. So as Angel said, our approach right now is to apply these genomic tools for the benefit of the growers and the cultivators and the breeders. We anticipate at some point there may be interest within unique groups, within unique growers and cultivators who may find us as a partner and work with us on customized projects or services. And that's the way things will evolve. That's my thinking process, okay? And that needs to come from them. So whether it is like working on higher density trichomes or more like the green revolution, shorter plants, quicker flowering with higher amounts of secondary metabolites, or the phytochemicals that we are talking about. It's possible.
Yeah.
And that's the scope. That's the next thing that is going to happen when we have more concerted effort on improving the crop quality and its productivity.
Very cool. Very cool. No, well, it certainly sounds, I mean, it's always an education, but, plant science is just, there's so much happening. And no, it's great to get an insight into what you guys are doing, and wishing you the very best of luck with it all. And thanks for coming on, and we'll speak to you again soon. Thank you very much, guys.
Thank you very much.
Thank you for taking time to talk to us. Pleasure to know you both.
You too. Thank you. All the best.
Thanks.
Cheers.
Thank you.